It's my pleasure to introduce today to Dr. Alexey Danilov, who has been an inspiration for the research in our lab. And well, let me just give some brief introduction. Dr. Danilov earned his medical degree in Russia and completed his a fellowship in hematology and oncology at the Tufts Medical Center in Boston, the US. He's a physician, but has a background in molecular biology and cancer cell signaling, as well as expertise in oncogenic drug development. Clinically, Dr. Danilov is a practicing medical oncologist specialized in the case of patients with CLL and lymphoma. He's now working at the City of Hope National Medical Center, where his research group is focused on the understanding of the B cell biology to find new therapeutic targets. As a director of the early phase therapeutics program, Dr. Danilov Group is evaluating novel targets in the ubiquitin-proteasome system and the oncogenic role of cyclin-dependent kinase 9. Building on his preclinical discoveries, he launched multiple early phase clinical trials with novel agents, and as a member of industry committees, he participates in the development of new drugs. He's also a co-chair of the Translational Medicine and Southwest Oncology Group Lymphoma Committee, and today he's going to talk about his discoveries of the use of the ubiquitin-proteasome system as a therapeutic target in hematologic malignancies. So, please.
Thank you very much, Carlos, for the kind introduction and for the invitation to be here. It's wonderful to be here in beautiful Santander. So I come from City of Hope, which was established as a tuberculosis hospital, but now it's a cancer center. It's one of the largest cancer centers in California, and certainly the largest one in Los Angeles area. So we are located in Los Angeles area, about 20 million people in between 4 counties. And we perform about 800 stem cell transplant per year, and have performed about 2,000 CAR T-cell procedures since we started. And our lymphoma group, which I lead, is a pretty large group of 15 physicians. And at any given time, we have between 40 and 60 active clinical trials in lymphoma. So we are a pretty large center when it comes to hematologic malignancies. As Carlos mentioned, I see patients, and I also direct a translational laboratory, which is focused on molecular therapeutics mostly, and identification of mechanisms of resistance and drug targets. And our group has focused on targets within the ubiquitin-proteasome system for a number of years. And we have worked on something called methylation, and that's how Carlos and I actually connected a few years ago. So just to give you a general overview of the ubiquitin-proteasome system, typically when protein needs to be degraded, it needs to be ubiquitinated by a ubiquitin-3 ligase. And this journey starts with ubiquitin activating enzyme, which is E1, and there are very few of these. And the most common ubiquitin activating enzyme is essentially UAE, and it can be inhibited by a drug called TAK243. On the other end of the spectrum, there are proteasome inhibitors such as bortezomib, ixazomib, and carfilzomib. So these drugs actually have made it into clinic. They are used in therapy of myeloma and lymphoma. They have official regulatory approvals in Europe and the United States. But by targeting proteasome, which is right at the end of this whole cascade, these drugs cannot achieve selectivity. So a bunch of proteins are being degradation is being prevented when you target proteasome. And like I said, on the other end, there is only one enzyme, essentially ubiquitin activating enzyme, which can be targeted by this compound called TAK243, which is still in clinical trials, but unlikely is going to proceed with much development. We published on that drug in lymphoma as well. There, we also have NED8 activating enzyme, which ultimately regulates kaolin ring ligases, which is one of the E3 ligases here. There are also successful drugs which target E3 ligases, specifically cereblon. And cereblon is modulated by lenalidomide, for example, also an agent which is used in therapy of myeloma mostly, but also lymphomas. So those are so-called immunomodulatory agents, but they're really E3 ligase modulators. And with modulation, what happens is NED8 activating enzyme actually modulates a certain subset of ligases called kaolin ring ligases. So while ubiquitin activating enzyme is one or two, there are a few of them. Then you have E2 enzymes. There are maybe a couple of dozens, but once you get to the E3 ligases, there you have about 400 or 500. And each of these have a fairly specific group of targets. So you can achieve some selectivity by targeting E3 ligases or modulating their function. So Carlos and I have been working on pivanedistat, which is actually an inhibitor of NED8 activating enzyme. And inhibition of NED8 activating enzyme changes the function of this kaolin ring E3 ligase and changes degradation of a certain subset of proteins. So this is work that we began probably 12 years ago, working with pivanedistat and trying to understand how it works in lymphoid cells. And as a model, we used CLL or LLC, chronic lymphocytic leukemia. So what you can see here is target is pivanedistat denidylates kalins, which are basically these moieties which need to be which need to be attached to the kaolin ring ligases for the kaolin ring ligase to be active. And by denedylating kalins, it blocks their function, and therefore you see accumulation of inhibitor of IKB alpha, phospho-IKB alpha, which is the negative regulator of the NF-κB pathway. So what we also have shown here is that this results in reduction of NF-κB activity in CLL cells, as examples here is P65 and P50, which are the NF-κB effectors. And that is accompanied by cell apoptosis. There is also a substantial dysregulation of the NF-κB signaling program, transcriptional program, with downregulation of many NF-κB targets. And we found that this apoptosis was actually dependent on the pro-apoptotic protein BIM. BIM is one of the NF-κB targets, and it also can be directly regulated by kaolin ring ligases. And what you see is accumulation of BIM in cells treated with increasing doses of pivanedistat. And basically what that does, it skews the balance between pro and anti-apoptotic BCL2 family members in favor of apoptosis, which results in caspase-3 activation and mitochondrial and intrinsic apoptotic mechanisms. And so if you actually knock down BIM in the cells, this results in partial cell rescue where they do not die as significantly. So pivanedistat induced apoptosis was dependent on BIM, at least partially. What we got interested, however, subsequently is the immunomodulatory functions of targeting nidylation. So nidylation, certainly there is a group of at least 40, 50 different proteins, how nidylation with kaolin ring ligases might work and be important in cancer. We never really saw complete apoptosis of CLL cells by direct targeting with pivanedistat. However, NF-κB pathway is a very important pathway in immunology, and that's actually where initially it was discovered. It was discovered as an inflammatory pathway, and it's crucial for function of immune T cells. So when we looked at T cells, stimulated naive T cells with CD328 and treated them with pivanedistat, we found that pivanedistat downregulated multiple T cell receptor-induced genes. And NF-kappa B signaling was one of the most downregulated pathways in T cells, which was actually downregulated very quickly within 3 hours. And you can see here that relay translocation is abolished when T cells were treated with pivanedistat. And initially, we started this work thinking that, well, we might see some pivanedistat toxicity in clinical trials, some negative effect on the immune cells. That was at least a hypothesis, but actually we saw in the end of it, we saw something quite the opposite. So what we saw is downregulation of immunosuppressive cytokines such as FOXP3 and IL-4, and at the same time upregulation of pro-inflammatory cytokines such as interferon gamma. There was also a minor upregulation of IL-17. Which sits in balance with FOXP3. So that was not entirely unexpected. However, the general production of IL-17 was very low. So basically what we found is that treatment with pivanedistat reduced differentiation of regulatory T cells and immunosuppressive T helper cells, the Th2 type. And at the same time, as a reverse of it, we found that there was increased differentiation of T helper 1 cells, which are essentially pro-inflammatory and can have anti-tumor function. So pivanedistat boosts inflammatory T cell differentiation. So with that information, which was collected in vitro, we actually went to the mouse, and we used the albumin-specific transgenic TCR CD4 T cells, transferred them into BALB/C background mice, and challenged these mice with albumin and toll-like receptor agonist with or without pivanedistat. So basically, these mice have this particular albumin responsive T cells that are being injected. And so that gives us an opportunity to track these T cells on a mouse and see how immune response develops. And so interestingly, we actually found a very similar result in a mouse as well. So we saw downregulation of FOXP3, so downregulation of regulatory T cells, and we saw it in both total CD4 population, which included the mouse own T cells, and we also saw it in albumin stimulated adaptively transferred T cells. So you can see that there was a reduction of Tregs in that population, which is KJ1 positive. At the same time, while we didn't see much effect on the interferon gamma in total T cell population, we actually saw an increase in interferon gamma in the immune-stimulated T cells. So we were actually, despite the fact that we knew we were also targeting NF-kappa B, we were not shutting down immune response. In fact, the immune response was substantially more active when mice were treated with pivanedistat. This is not something that we necessarily expected. So we proceeded with additional experiments trying to understand how targeting edulation might impact anti-tumor immunity. And so what we did here is we took an A20 lymphoma model, which is a syngeneic mouse model where you basically take mouse A20 cells and inoculate them into the flank, and that allows you to study tumor-infiltrating lymphocytes in the mouse. And when we treated these mice, A20 inoculated mice, where's my mouse, with pivanedistat, we actually saw a partial reduction in tumor growth. You can see here the difference is not great between the black and the red line. The red line is pivanedistat treated mice, and the black line is control mice. The difference is not very big, but there was some reduction in growth of of the cells. And at the same time, there was a reduction in tumor weight. But importantly, what we found is there was enhanced T cell infiltration. You know what? Do we have? No? Okay. There was enhanced T cell infiltration in the tumor microenvironment. You see that there were more CD4 positive T cells in the pivanagystat treated mice down on the graph, where pivanagystat is in gray. And there was also more infiltrating CD8 positive T cells per milligram tissue in pivanagystat treated mice. And importantly, these T cells, particularly CD8 T cells, were also expressing interferon gamma, meaning that they were actually activated in the tumor tissue. So pivanagystat treated mice in gray, there was at least triple the amount of interferon gamma secretion by the CD8 T cells. So, then we became really interested. A20 cells actually are resistant to pivanagystat. So if you take it A20 cells, treat them with pivanagystat in vitro, they actually do not die. So we started thinking, why do we have this reduction in tumor growth in pivanagystat-treated mice, whereas the primary cells are actually completely not responsive to pivanagystat? So it is possible that the immune effect is fully responsible for that. So we decided to take out this immune effect by using an anti-CD8 antibody. And basically what happens is if you treat mice with anti-CD8, then it completely abrogates the effect of pivanagystat. So what you see here is the anti-CD8 treated mice in blue, the vehicle control is in black. And you can see that if you treat mice with anti-CD8, the tumor actually grows a little bit faster. So if you compare, the top line is blue, the black line is your control. So the tumor begins to grow a bit faster. Then if you treat with pivanagystat, it's in red, it grows a bit slower. But when you combine pivanagystat and anti-CD8, the effect of pivanagystat is completely abrogated. So essentially, the tumor grows as fast as with anti-CD8 treated in anti-CD8 treated mice. So basically, this gave us a suggestion that the effect of pivanagystat in this model, at least, is fully immune mediated. And then, if you the other way to check this was actually to knock down UBC12 completely, which knocks down the whole methylation pathway in a cell. And so this is what we did here. And essentially, what happens here is even when you knock down the methylation pathway in the A20 cell, so pivanagystat has zero effect on the A20 inoculated lymphoma mouse cell, you still see this effect. So if you compare the red line, which is the A20 cells where methylation pathway was completely knocked out, and the efficacy of pivanagystat in vivo is the same as those which do not have the methylation knockout, which is the black line. So basically what this shows here is that you take the A20 cells, completely get rid of the methylation pathway within your tumor cell, and still you see efficacy of pivanagystat in vivo. So that again suggests that we are dealing with immune-mediated effect rather than direct effect on the tumor cell. And what was important also is when we combined this with anti-PD-1 antibody, there was very strong synergy. So the growth of the tumor essentially stopped completely once you combined pivanagystat with anti-PD-1. So that's on the right side. You see again, the control is your black. And the green line is the mice which were treated with anti-PD-1 antibody and pivanagystat, and that line pretty much flatlined and for as long as we kept this mice. So those tumors stopped growing. So basically what we felt is that in at least in fairly immunogenic lymphoma model, which A20 is, and the effect of pivanagystat targeting methylation mainly works via immune modulation, not so much via direct effect on the tumors. So nevertheless, we wanted to run a certain clinical trial, and we wanted to combine pivanedistat with PD-1 in lymphoma, but Takeda didn't want to do that. We also had some preclinical results in a combination of pivanedistat with ibrutinib in CLL model. And so we initiated that trial, which Takeda was much more interested in for whatever reason. So we started the phase 1 investigator-initiated study, and at the first step, we needed to find the right dose for pivanedistat in a human. So this was the first in human combination of pivanedistat and ibrutinib, which is a brutonizing kinase inhibitor used at a standard dose. Pretty standard inclusion criteria, and we essentially designed it as a toxicity study. Our recommended phase 2 dose was 50 milligram per meter squared of pivanedistat, which was the maximum dose that we used. So we actually never really reached the maximum therapeutic dose. There were some standard adverse events, which were mostly attributed to ibrutinib, which was bruising, diarrhea. There was one interesting case which we couldn't explain with mediastinal hemorrhage. Again, we attributed ultimately that this was because of ibrutinib, because pivanedistat has never been known to cause bleeding, and in no studies this has occurred. The efficacy, however, was a little bit underwhelming in this patient population. It seemed to work the best in mantle cell lymphoma, where there was 100% overall response rate with this combination. And I still have one patient who stopped pivanedistat, but is continuing on ibrutinib 5 years later on the study, and I still cannot close the study. So anyway, at least what we have shown is that pivanedistat was safe in combination with ibrutinib in patients with lymphoid malignancies. And we also collected some immune cells from these patients, we collected some blood cells in the process, and have found that there was increased interferon gamma signaling in CD4 and CD8 T cells obtained from patients treated with pivanedistat, essentially mimicking our results that we saw in vitro and that we saw in a mouse. Again, suggesting that the immunomodulatory effect of nadylation is very important in this case. So this is so far where we stopped with our nadylation studies. Ultimately, what we have found is that NAD8 inhibition induced apoptosis in CLL cells, mainly by blocking the NF-kappa B pathway. We also had a couple of side projects where we looked at DNA damage response. NAD8 also regulates a pathway called CDT1, which controls DNA replication. So that's much more important in rapidly cycling cells. CLL cells do not cycle so rapidly. So, what we also found is that, which I think is more important for at least our model, is that NAD8 inhibition reversed the immunosuppressive phenotypes in these models and augmented inflammatory T cell polarization and enhanced the function of tumor-infiltrating lymphocytes. So how exactly that happens, we still are not entirely sure, but we actually think that this happens through the hypoxia-induced factor alpha, HIF-1 alpha mechanism, because when we actually did a few experiments, didn't go too far down that road, but when we knocked out HIF-1 alpha in immune cells, we actually were able to abrogate these nadylation effects. So we do believe that this effect on inflammatory T cell population is HIF-1 alpha mediated. So this is with the pivanedistat story that maybe we'll talk more about it after we are done here. But in the second part, I wanted to talk a little bit more about cereblon as a target, and there is a whole emerging field of degraders. So and again, this particular mechanism still sits in the ubiquitin-proteasome pathway. If you remember the original schema where I showed the whole UPS, there are E3 ligases, and we were just talking about Carling ring ligases, which are regulated by nadylation. But there is another group of ligases called cereblon and von Hippel-Lindau ligases. So quite a few E3 ligases whose function is to label proteins for degradation with ubiquitin. So the proteolysis targeted chimeras are drug molecules which essentially were developed over the past 30 to 40 years. So this is a relatively new field. So each chimera consists, or a product shown here on top, consists of a hook, which is a ligand for your protein of interest. On the other end, there is a E3 ligase ligand, which is a harness, and they're linked by a linker. So you see the blue triangle and the red triangle linked by a linker. That's your proteolysis targeting chimera or a degrader. And so what happens is when the product that links your protein of interest, on one hand, on the other side, it invites the E3 ligase, and that results in ubiquitination, forced ubiquitination of the protein of interest, which then goes through degradation by the proteasome. And it's a really interesting mechanism because this one molecule can go back into circulation and in the cell and attack more and more targets. So if you have an oncogenic protein, which is your target, you technically only need one degrader molecule in a cell to destroy all of your target kinases or whatever they might be. So it is possible here that you do not need high concentrations of the drug in the blood or in your tissues because the drug gets recycled. And that is not a frequent thing. You know, chemotherapy goes into DNA, damages it, and then it gets destroyed. Kinase inhibitors need to stay linked to the kinases of interest, so you need certain concentration of this. So this, it's a very different mechanism than anything else. So the other aspect of it is that you can target multiple kinases. For example, those are the ones listed here that are being investigated as product targets like BTK, obviously, BRAF, KRAS, and many others. You also can target non-kinases, transcription factors. For example, STAT degraders are out there now. You can target receptor proteins. Pretty much, you can target anything as long as you can find where your chemical will bind to the target. One of the most elusive targets in oncology is MYC oncogene, right? So MYC protein. There are no good drugs which target MYC. So there may one day be a degrader which would degrade MYC. Of course, the problem with MYC is that it doesn't have a good surface for any drug to attach. And that's why nobody has successfully designed a chemical against it. But I'm sure that eventually it will happen. Also, on the other side of the link, where you have a harness which targets to a certain E3 ligase, you can basically invite an E3 ligase of your choice. And cereblon used most frequently now. It's the same target as lenalidomide, but you can also use von Hippel-Lindau, MDM2, inhibitor of apoptosis, and other E3 ligases. And that will confer certain tissue selectivity for your drug also. So the products have been developed for the past 30 years. There has been multiple Arvinas trials, which were actually focused on androgen receptors. So this was the first target being investigated in clinical trials with androgen receptor degraders. There was a publication in New England Journal of Medicine just about 3 months ago, a first randomized trial with a degrader in breast cancer, estrogen receptor degrader, where it was compared in estrogen receptor hormone therapy resistant breast cancer. The degrader was compared to fulvestrant and resulted in improved progression-free survival. So this was the first positive phase 3 study for any product in oncology. So these drugs, I believe, will see more and more studies like this. And so this is the target. The target here is estrogen receptor, but also you can, like I said, you can target many other different things. For example, BCLX is a very attractive target. DT2216 is BCLX degrader investigated in preclinical models in acute myeloid leukemia. This is work from Marina Konopleva, who used to be at MD Anderson, and now she's in New York. And she used the AML model, which evolved from myeloproliferative neoplasm, which is JAK dependent. And basically, with this BCLX degrader, she found that it works in primary AML blasts, which is the top left figure. It also synergizes with azacitidine, a drug we often use in treatment of AML, which is the bottom figure on the left. It shows BCLX degradation in the cells, which is the top of this middle Western blot. You can see that BCLX disappears when cells are treated with DT2216, and it also slows growth of acute myeloid leukemia in a mouse xenograft model, which is on the right. The blue line is DT2216 treated mice. And in these mice, you can also see degradation of BCLX cell. Another target is BCL6. This one is in clinical trials. There is actually very good data emerging in follicular lymphoma, diffuse large B-cell lymphoma. And at City of Hope, we have two different BCL6 degraders now in clinical trials. And this was the first data with BCL6 degrader presented in Lugano in the summer. So these were patients with diffuse large B-cell lymphoma, follicular lymphoma, which were treated with at least with four different prior therapies, so heavily resistant patient population. And with this BCL6 degrader from BMS, Bristol Myers Squibb, you can see overall response rate was 80% and complete response rate is 24%, which is actually a very good result for this heavily refractory patient population. And what you see on the graph on the left is rapid degradation of BCL6 essentially at every dose of the drug. So what my group hasn't been involved with is BTK degraders. Of course, I mostly see CLL and mantle cell lymphoma in my clinic. And in CLL in particular, mutations in Bruton tyrosine kinase are very important because they confer resistance to BTK inhibitors. The most popular one is C481S, when cysteine is replaced with serine at the 481 residue, and this confers resistance to all known covalent BTK inhibitors, which we frequently use in treatment of CLL now in ibrutinib, zanubrutinib, and acalabrutinib. And there are some other mutations there as well, like, for example, L528, which will also confer resistance to non-covalent BTK inhibitors, such as pirtobrutinib. So it is certainly a very attractive option to degrade BTK, because then you don't care what kind of a mutation BTK might have. Another positive about degrading BTK is BTK actually might have a scaffold function, where it can propagate the signal of the B-cell receptor, even if its kinase function is disrupted. So what you can see here with certain mutations, you see if you look on the phospholipase C gamma 2, you see it gets activated with wild type BTK, it gets activated with C481S mutant BTK, and concurrently, you see activation of phospho-AKT, right? So if you look at 2 lanes down. But there are certain mutations, for example, the one I just mentioned, L528W, this is the one almost, the penultimate one, almost the last one. You see there is no activation of phospholipase C gamma 2. It's blank there, but you still see activation of AKT, which is the downstream target. And so we believe now that this is because of the scaffolding effect of BTK, where BTK, even when it's not activated, it complexes with other kinases in the B-cell receptor complex, and the signal is still propagated, the cell can still survive. So that's why BTK degradation is also very attractive overall. So we worked with 2 cereblon recruiting degraders. One is NX-2127 and one is NX-5948. And they are different. They're different in the way how they actually approach cereblon. So NX-5948, which is in red on the left, It degrades BTK, but it does not modulate cereblon. And NX-2127 is also known to modulate cereblon, meaning it works just like lenalidomide. It actually creates um, um, changes the property of cereblon, where it acquires new capabilities to process certain near substrates like ILS and Icarus. So cereblon changes its affinity for these new targets and degrades and results in degradation of ILS and Icarus. So this is how those two drugs are different, and I'll explain a little bit more about that. It's interesting that all these BTK degraders that we have are very selective. So here is mass spec on NX-2127, and what you can see in the graph below, there is only BTK that it disappears from NX-2127 treated cells, and very similar data with NX-5948. So even though the warhead for both drugs is actually ibrutinib, which is not a selective kinase inhibitor, right? It has thousands, potentially targets, well, at least few dozens of kinases that it inhibits. But once it gets bound to a linker and a harness, it becomes very selective. So both of these degraders are highly selective in the proteins of interest that they target. So we treated CLL cells with these drugs, NX-2127 and NX-5948. And what you can see here is that both drugs degrade BTK. So you see DMSO-treated cells, there's BTK once you get the drugs, even at lower concentration, 100 nanomolar, BTK is obliterated completely, it goes away. With NX-2127, which has the immunomodulatory properties, which modulates cereblon to impart these neosubstrate qualities, you also see degradation of ILS and Icarus. You do not see that with NX-5948. So both drugs outperform pirtobrutinib against mutated BTK. What you can see here is that if you introduce mutated BTK into the cell, the cell still cannot survive when it's treated with the degrader, but it certainly can survive when it's treated with ibrutinib or pirtobrutinib. And so the next question, so we have two different degraders. One is a clean BTK degrader, and the other one has immunomodulatory function. And so we ask the questions then, what are the effects of these degraders on T cell functionality, and is this potentially important in anti-tumor effect in lymphoma? So we show here that in T cells as well, ILS and Icarus are also degraded by NX-2127, but not by NX-5948. This is what's shown in the graph here. What we see here is that NX-2127 downregulates very few pathways in T cells, but actually instead triggers interferon gamma and TNF alpha gene signature in primary sorted CD3 naive T cells. And so when you do gene set expression on GSEA, you see upregulation of hallmark inflammatory response, interferon gamma response, and TNF signaling in these cells. So there is clear effect of this immunomodulatory degrader NX-2127 on the pro-inflammatory pathways in T cells, which can also be linked to anti-tumor response. And when we checked by flow cytometry, we also found that interferon gamma is upregulated by NX-2127, which now has a name of zolibrudomide, but it's not upregulated by NX-5948, the clean BTK degrader, which now has a name of bexabrutideg also. Again, same data in polarized cells. If you take Th1 conditions and culture T cells under these conditions, they have more propensity towards becoming pro-inflammatory immune cells. And what you can see that NX-2127 enhances this by enhanced secretion of interferon gamma and IL-2, and you do not see an effect by NX-5948. There was also some effect on Tregs as well. And most importantly, we found that just like lenalidomide, NX-127 also enhanced synapse formation to levels comparable to lenalidomide itself. So in the middle graph, where the white arrow is, you can see that there is a kissing between the CLL cell, which has a red rim, and the T cell, which has a purple rim. And you only can see that with NX-127 in the middle and lenalidomide on the right. You do not see that with the ibrutinib or NX-5948. And so consistent with that, we also saw that the NX-127 enhances T-cell mediated killing of lymphoma cells. In this case, we used OCI-LY19 cells as our target cells. And you can see in red that the NX-127 it results in 20% more cells killed by T cells in a cytotoxicity assay compared to other drugs. And this was also accompanied by increased secretion of granzyme B by CD8 T cells. So, encouraged by this work, we looked at NX-127 treated patients, and first we used bulk RNA-seq, and we found that non-responders exhibited higher Th17, Th2 gene expression levels compared to responders. But bulk RNA-seq, of course, you have a lot of dirty cells there, so we went to single-cell RNA-seq immediately. And found that in non-responders, exhaustion markers, CD8 exhaustion markers were upregulated compared to responders. And in the meantime, in the responders, we saw increased activation and increased oxidative phosphorylation in CD4 T cells, again, in those patients who had fast response to NX-127. So there is clearly at least some component of immune modulation that is important in clinical efficacy of these drugs, of the BTK degraders. So overall, BTK degraders are doing really well in the clinic. There are a couple of molecules now which are pretty far advanced, which were tested in highly refractory CLL, which progressed on ibrutinib and venetoclax, and responses are very high, essentially in excess of 80%. And these drugs are very well tolerated, with most adverse events consistent with what we see with the ibrutinib, such as some minor bruising, neutropenia, and no bad toxicities at all. And in vivo as well in patients, these drugs degrade BTK very quickly, regardless of which mutation is present, whether it's wild-type BTK or C481S or any other mutation. There is very rapid degradation of BTK by these drugs, by BTK degraders, which is essentially the whole proof of concept of all the preclinical data that has been generated so far. And also, interestingly, that efficacy of these drugs is very good across the whole spectrum of different mutations, whether patients have mutations in BTK or other concurrent high-risk mutations such as P53, Notch1, SF3B1. All these patients respond to treatment with BTK degraders. So the conclusion for the second part of my talk is that proteolysis-targeted chimeras is a novel molecular approach, and we will see more and more of these drugs in the near future. And I've talked to you about BTK degraders, which also can have some T-cell immunomodulatory effects. We are still figuring out how important that is actually. We will proceed with some mouse work, and it would be interesting to combine these drugs with, say, CAR T cells and bispecific antibodies. And BTK degraders do show very good efficacy in clinical trials with good safety profile, and randomized phase 3 trials are being planned with these drugs to seek regulatory approval. So thank you very much, and thanks to all the patients who participated in the clinical trials, and to my laboratory, which is pictured here. Thank you. Happy to answer any questions. Thank you. I can start with I'm touching, so I'm going to
I'm going to do some B cell physiopathology question. Where do you think this ubiquitin-like modulation, postulational modulation is at which level of the biology of these CLL cells is located? I mean, there's several processes that are that we know now that are working in these CLL cells, you know, like overexpression of these E1F or A1 and some others, no, and the BTK and the BCR receptor overactivation. Where do you think this methylation, this ubiquitination process is in this hierarchy of events?
Yeah, so that's a very good question. I'm not sure I know the answer to that question. I think so. We actually do not know what regulates NED8 activating enzyme activity in CLL cells. So there's not too much known about it in any tissue type, not just CLL or lymphoma. It is something that's a fairly ubiquitous process. So it's not a process that is unique to CLL cells, but methylation seems to be more activated in CLL cells compared to, so say, non-cancer cells. So if I had to guess, it probably has to do something with B-cell receptor signaling, but I don't know for sure.
Thank you very much. It was very interesting and very comprehensive. So, but well, I work in myeloma, okay, and we have tested just pevonedistat, trying to add to that in myeloma. And well, I think that the data with pevonedistat as single agent is mild, it's not very important, but I think it's compelling the data with the enhancement in the immune system. So you have shown the data with the PD-1 inhibitors that it really has synergistic effect. What do you think? Would it make any sense to combine that with the new immunotherapeutic approaches, bispecifics? I don't know if CAR-Ts, but maybe this is something that could work. Or do you have models? Have you tested that, or do you think it could work?
Yeah, no, that's a good point. So actually, when we first approached Takeda, the trial that we wanted to run was pevonedistat plus nivolumab in refractory Hodgkin lymphoma. Because for a lymphoma doc, Hodgkin lymphoma is very immune active disease, right? So nivolumab has single agent activity, so that's where we wanted to be. But Takeda wasn't interested. They said Hodgkin lymphoma, there are no patients. So we ran a CLL study instead, which is fine. We started work with pevonedistat and CAR-T a little bit. So right now, pevonedistat is not being developed by Takeda. There are some small companies which occasionally are interested. So we have not designed the next trial, and we haven't taken our CAR-T work too far. We saw a little bit of enhancement of CAR-T cell function with pevonedistat as expected. We haven't tested the bispecific, but I agree with you. So actually, pevonedistat with bispecifics would probably be most interesting, and it's a pretty safe drug if you use it at a lower dose. The only thing, it's administered inconveniently. It's intravenously. Pretty frequent intravenous infusions, like I think it's day 135 on 28-day cycle. So that's the only inconvenience. If we could formulate it orally, that would be much better. But yeah, I agree with you. Something like bispecific combination would be really interesting.
Two more questions just quickly. One is, how do you explain the activity of the products, the products that are really degrading the BTKs in BTK mutated cell lines or in BTK mutated cells? Because in this case, you have constitutive activation of the BTK. So it's just that you are reducing the amount of BTK, and then there is this.
Yeah, so but it does degrade mutant BTK as well.
Okay.
Right. So so the non-covalent, so pirtobrutinib does not inhibit certain, or ibrutinib does not inhibit BTK-C481S, but the degraders, they bind outside of the ATP outside of the region where kinase inhibitors bind. So it's not ATP competitive, so it binds outside that region. I don't know exactly why, it's all proprietary, right? But it degrades essentially any mutation in BTK will be degraded by these degraders. However, there was at least one small publication which suggests that there are certain mutations in BTK which emerge while you're on degrader that reduces the binding of the degrader. But that would be potentially mechanism of resistance to the degrader. But so far, any of these mutations we know, they are all susceptible to BTK degraders.
Okay, perfect. Don't worry, I will not ask anything.
Questions? Well, maybe we should cut here because we have to go to work. So thank you very much again for your nice talk. And si alguien está interesado en reunirse con el doctor Danilov, alguien de algún servicio, vamos a estar hoy por hematología. Así que, bueno, pues contactáis con nosotros y podemos organizar una reunión o lo que sea. Bueno, gracias por la asistencia. Thank you.